Good. Hello, everybody. My name is Andy Holt. I'm the Chief Commercial Officer for Encardia. Everybody's just filtering in here, but hopefully you can see our four esteemed panelists to talk a little bit about neurodegenerative disease and development today. It's just right at the top of the hour, so we are going to give a minute or so for folks to file in. In the interim, I'll say this now and probably remind everybody of it. As we go through, you're more than welcome to ask questions of the panelists. We'd ask you to do so through the Q&A button if you're not familiar with the Zoom webinar tools. It's at the toolbar at the bottom of the screen. That Q&A will allow your question to be submitted in writing to both myself and to the panel, so we can incorporate it into the flow of the conversation. So if you can, just keep an eye on that Q&A button when something strikes you, so throw it into the chat through that, rather than through the chat button, where we're not going to be monitoring as much. And then we can make sure that your voice and your question get heard here and addressed by our panelists. So again, we'll just give another 30 seconds or so. I see lots of folks are still filtering in. So yeah, we'll just give it another minute and then we'll get this show on the road. So really grateful for everybody's attendance here today. All right, I think we're 2 minutes past, so I'll get one more set of reminders out for everybody, and then we'll get this going. Again, my name is Andy Holt. I'm the Chief Commercial Officer for Encardia. I'm going to moderate the panel, which means I'm going to try to stay out of the way as much as I humanly can. As a quick reminder for anybody who's just joined, we are happy to accept questions via the Q&A button in the toolbar on the bottom of your Zoom screen. And with that, I'll do a quick round of introductions of our panelists, and then we can get on with our discussion. So, Dr. Gilbert, would you mind going first, introducing yourself to the audience here?
Sure. Thank you so much for having me. My name is Rebecca Gilbert, and I am Chief Scientific Officer at American Parkinson's Disease Association, which is a nonprofit organization which works to support people with Parkinson's disease.
Thank you so much. Shushant, would you rather, you want to go next for us?
Yeah, certainly. So hello, everybody. My name is Shushant Jain, and I'm the Director of Discovery Technologies at Encardia, where we leverage iPSC technology to help build translational disease models to facilitate the drug discovery path.
Dr. Ross?
Hi, I'm Dr. Ross, VP and Head of Neuroscience at Immuneering, and I focus on Alzheimer's disease.
Astrid, do you mind bringing us home?
Sure, thank you for having me here. My name is Astrid Vallès, Director of CNS Disease Biology at Unicure, gene therapy company based in Europe and in the US. We focus on liver and CNS diseases, and in the CNS we have programs in Huntington's disease, but also Parkinson's disease and other neurodegenerative disorders. I'm looking forward to the discussion today.
Well, I'll just say on behalf of Encardia, on behalf of the whole group that's come here to listen to your thoughts today, really grateful. I probably told the group here too much that I'm really excited to hear both the diversity of clinical and scientific experience, as well as the diversity of disease focuses that we have here. Hopefully gives a pretty broad perspective, both engaging with patients, engaging in development, and then just engaging with the pure science of the challenges here. And I think that's really where I wanted to start the first discussion point for us to help stage set a little bit. You know, we've known about Parkinson's since 1817, Huntington's since 1872, and the first case of Alzheimer's was diagnosed in 1906. Your professional lives, pardon me, here on the panel have really been focused on addressing those diseases. Why has this been so hard, do you think? And Dr. Ross, I'll ask you to go first, and then I'd love to hear from the entire panel as we go around. But you know, for things that have been around that long, we're still in some ways at fundamental understanding of why the disease exists at all and how it works. Can you share your perspective on what makes this such a steep hill to climb?
Yeah, particularly with Alzheimer's, you know, I think part of it sort of just been sort of the evolution of our understanding of it. So, you know, you go back to the 1970s, this is like sort of the first time for which we started to subgroup out dementia into different diseases. So before then, it was just sort of considered an age-related thing. You got older, you started to lose your memory, et cetera, et cetera. But through sort of investigations of neuropathology, it became very clear that dementia is actually made up of different sort of diseases, like Alzheimer's being sort of the more frequent one. But also you had also the Lewy body as well as frontotemporal. So we got a better understanding over time of these diseases. But having said that, I think one of the things in particular with Alzheimer's is that it's a very complex disease, right? And it shows itself in many forms. I see patients every so often, and each patient is very different in terms of their level of progression, their speed of progression. And so really, I think part of it is just sort of the complexity around diseases, as opposed to sort of, for example, these monogenic disorders, like for example, in cancer, where you can have sort of a single point mutation, and that's enough to one, identify the patient, but also sort of predict their prognosis, where most of the cases for Alzheimer's, you really don't have that genetic basis for understanding. So it's definitely been a challenge. And the last thing I'll say is biomarkers. For Alzheimer's, Parkinson's, ALS, we really don't have the sort of tools in terms of markers that other diseases, sort of like cardiovascular disease, any of those indications. We really just don't have that yet.
Thank you. Rebecca, do you want to share your perspective more from the Parkinson's side of the world?
Yeah, sure. So I, agree with everything Dr. Ross said. Those are all incredibly difficult challenges that are unique to the brain. And I'll add one, which is that by the time we intervene, or we have intervened in clinical trials, there has been so much neurodegeneration that has taken place already, because these diseases are extremely slow. They start really sort of unbeknownst to even the person with Parkinson's, that things are developing in their brain. And by the time something is noticed by even the family or the person themselves, there's been so much destruction that we're not really sure whether we have the tools. We don't have the tools right now to do anything. So that brings the challenge of identifying people earlier on, back to Dr. Ross's point about biomarkers, finding something that you can detect early enough so that you're really at the start of the neurodegeneration and have a hope of intervening with success. So I think that's a huge challenge as well.
Shusant, maybe you could go next. We've heard folks already mention kind of tools and targets to address. From your work in trying to develop those tools, do you see similar complexity from kind of trying to recreate?
Yeah, I think I'll pick up on the point that Ross and Rebecca made in terms of just the heterogeneity of these diseases. I mean, for a lot of the neurodegenerative, there's some aspect is driven by a monogenetic component, but indeed, large proportion is there is still a genetic basis for a predisposition for a lot of these diseases, but the number of pathways that are involved and potential mechanisms is enormous. So really being able to dissect out who is, in which individuals, which pathways are contributing the most to disease continues to remain a problem. And I think, if we go back to the original statement you made, Andy, in terms of when the disease was identified or described, and then ultimately where we are today, I think the next generation technologies, and I use NGS here as an example, is really helping us to begin to understand which subpopulations, or even further delineate Alzheimer's disease into further subcategories. And I think that is also going to be an important part in not only understanding which biomarkers are best for subgroups of individuals, but also then which individuals are best suited for which clinical trials.
Now, Astrid, I'm curious, given your experience where there's multiple variants of a genetic disease that you're quite focused on in Huntington's, how you hear that sort of combination of pathway and monogenetic root causes and the toolbox that you're bringing to this, whether you feel like there's chance for acceleration or you're just going to NGS everything and find more than you thought and end up in a spot that's more complicated than you started with.
Well, yeah, I cannot agree more with everything that has been said. And actually, in the monogenetic disease space, you'd think on paper that it's very simple, but it starts getting a little bit more complicated the more tools we apply and the more data we generate. So in the case of Huntington's disease, monogenetic disorder, autosomic dominance, we know the target, right? This is easy, it's Huntington. But we know that genetic makeup of the patients make actually the patients very diverse. And as we learn more about this, we know in the case of Huntington's disease, is a mutation in the Huntington gene, there is a CAG repeat expansion. But depending on the length of this expansion, you also have a more faster progression or less fast progression, depending on the genetic makeup on other genes or other single nucleotide polymorphisms. You have also an earlier or later disease onset, which has brought new targets actually that are targetable, I think are targetable for Huntington's. And I would want to bring to the table the possibility of addressing multiple targets at the same time and multiple mechanisms in order to be more efficacious. So I completely agree with the window of opportunity, which, it's a challenging in diseases that have not such a strong genetic component. But even in the case of Huntington, is a challenge. When there is no cure, do these patients want to test themselves or not when they know, there is nothing for them in the avenue? And if they do, still, they're 18, about their 40s, they expect to have they should have some manifestations of the disease. If you would start an early trial, how would you know that you are being efficacious without the right biomarkers, without the right readouts? So this is the level of complexity that we are dealing as well in the case of monogenetic diseases. And I could talk a lot about this, but maybe I want to bring it back to some of the panelists here as well.
No, thank you for that. I think it sounds like a combination of genetic and non-genetic with no biomarkers and tools to chase. So maybe that's the answer of why it's so hard. Almost more of a philosophical question I was interested to hear you guys react to is, do you think are these diseases an order of magnitude more complex, or are they just getting more attention right now? And it's a little bit, Ross, what you were saying about in the 70s, it was just getting old and losing your memory. And now there's 16 different variants of each of these sort of diseases and getting more and more understood. Do you, maybe Rebecca, it's best to start with you as you're on the advocacy and kind of fundraising side to think about, do you see a change and shift in focus? Obviously with Parkinson's, there's a number of really notable patients in that particular disease, but how do you feel, is this a societal reaction because we're all getting older and we don't want that to happen to us? Or is there really something, are we at a moment in time here that's unique?
So I think a combination of those things. I think certainly, people are expecting a better aging than perhaps in the past, and they should. We all should. And moving forward in society, we want to improve. And it isn't acceptable to be 80 and to lose your memory. That should not be something that we accept. And the same thing with Parkinson's disease. It's a very similar thing. People would get a little stooped, they get a little slow, they shuffle a little bit. Well, that's what happens. But no, it isn't what happens. And we really want to do our best to undo those processes. So I do think we're less accepting, and that's a good thing. And I also think that there is more of a political will. Because people are less accepting, there's more of a political will. And I should mention that currently there's the National Plan to End Parkinson's Act, which is going through Congress right now, very similarly modeled on an Alzheimer's bill that went through Congress and led to a lot of increase in funding for dementia and various dimensions, including Alzheimer's. So a very similar thing is happening now in Parkinson's disease, and that's really taking the patient population and advocacy by storm, really elevating that feeling like, yes, we can do this and we can intervene, and our government is behind us. So I do think it's a moment. I do think that it's a good moment in history to move this forward politically as well. So I'm hopeful.
Thank you. Astrid, I'd be curious, in Huntington specifically, but just in your work in I don't want to make you answer for the healthcare environment in Europe, but you and Shui are going to have to here on the panel for us. Do you see a similar sort of frame shift, or is a similar increase in focus and attention on the work that you've been doing, or do you think this has been just kind of a slow progress? Like, I guess, what's the slope of the line from your perspective? Does it feel steep or kind of progressive?
Yes. So I will say from the rare disease space, it's very specific to the indication and very often to non-for-profit organizations and funding, from externals that will determine where the interest or the resources in a particular indication or disease. In the case of Huntington's disease, we are very fortunate that there is a broad network both in Europe and in the States supporting this research. When it comes to specific governments in Europe, as you know, Europe is a combination of different lands. I would think this is specific for every country, and it's difficult to speak for all of the countries. But I don't know, Shu, what you think about this and how you see that.
Thanks, Astrid.
Sorry.
No, that's okay. Yeah. Again, I wouldn't be able to comment on the on each country, but personally, I do think there's more of an appetite, both from academia and from the for-profit entities, to engage with each other and begin to tackle these diseases. And that is both from the basic research perspective, but linking that through to therapeutic development. And because of the increased collaboration between these experts, I think there they have greater confidence that we are getting in a better, or we will be in a better situation to be able to start tackling these.
Go ahead.
And maybe I can pick up as well. I think it's a very good point that you make, Shu. And there is collaboration between different countries, and of course, EU funding that supports huge networks, indeed, which can go in the spectrum from patient organizations to caretakers to biotechs, academia, all the spectrum. And I think this is a way forward, and I'm very happy to see that there are more and more of these consortia that are supported by EU grants, and having the whole spectrum of people working on this, it's what can make a change as well. So I would agree very much with what you pointed out, Sure.
And Andy, you put it best with, in terms of a sort of a frame shift. I really like that term because, back in 1906, when Alzheimer's, first, sort of diagnosed a patient with Alzheimer's, the life expectancy for Americans were, sort of like late 40s, early 50s, which will put you sort of in that, ballpark of being, maybe having early onset Alzheimer's if you had the mutation. But the 20th century, sort of led this sort of medical revolution with different medicines and increased our life expectancies into our 70s and beyond. That we're now seeing sort of more and more cases of folks with dementia or Alzheimer's. And, from a sort of a healthcare standpoint, we know that currently in 2022 here, we're spending about $355 billion taking care of these people, not treating them, taking care of these folks. And by 2020 or 2050, it's going to balloon all the way up to $1 trillion. So we sort of have sort of an invested interest, as taxpayers and caregivers to find a solution for these folks, because it's starting to have sort of a financial burden on society.
I think that's an interesting way to put it, and maybe a heavy weight for any company or one project to try to bear, right? But it is, I think a lot about like the cost of genetic medicines versus the incredible therapeutic benefit they offer. But when you reconcile it against palliative care and decades of memory care or ambulative care or all those sort of things, it starts to seem a little bit cheaper. Maybe to get a little bit deeper into, so if we understand that it's hard and why we're getting attention, to start looking at what's been going on once these therapeutics hit humans for the first time, and kind of the fits and starts that we've seen there. And Ross, I may start with you, just because Alzheimer's gets all the news, and you volunteered to be our Alzheimer's person. When we look at clinical trial setups and designs, for these sort of diseases where we've already heard the therapeutic window is hard to identify and restorative care may be impossible, how do you reconcile the sort of research and then the clinical results of that research in recent news in Alzheimer's, the Aduhelm and then the newer set of drugs that are coming through? Do you see that as kind of the price of progress, or do you see that as areas to really take learnings from and do things differently with the next set of shots on goal?
Yeah, I think with every clinical trial that we run in AD, we're learning something new. And I think one of the things that I sort of always have a gripe with, or I think is a challenge with clinical design for these patients, is what is our primary endpoint, right? If you take a look at any given trial, it's been very inconsistent as to what memory tests that they're using to see or measure a clinical benefit. You have things like the clinical dementia rating scale, which is the CDR. You have mini mentals. You have ADOS-COG, or sometimes you have a combination. And for a lot of these patients, when they actually do go to their PCP, they're not given a CDR. They're usually given something that is pretty easy to administer, like a mini mental. So the question becomes, what standard do we hold ourselves to sort of measure progress in the clinical sort of environment, in the trial environment, that will translate to everyday use for PCPs out there that have to be sort of charged with, one, diagnosing these patients as well as treating these patients. But even, let's take a step back before that. How do we even sort of measure whether or not a particular therapeutic is going to be efficacious once we even dose the first patient? And that challenge is really in the lack of really good and robust animal models that we have for these indications. And for Alzheimer's, there's probably over 20 different amyloid models to study amyloid, but we know that amyloid really doesn't, at least to date, really move the needle for a clinical benefit. So we really need to invest more into understanding what models can be very predictive and sort of translate to the clinic.
Well, and Shui, maybe I think that's an interesting question to get your input on when we talk about that sort of connection and translation problem. The Modernization Act just passed saying that you don't necessarily need animal models at all. So how do you, we'll see, but how do you reconcile what you just heard from Ross on 20 different animal models and no successful clinical trials, basically, with effective drug development after because you've probably seen most of these usual suspects come through your hands at one point or another. help us through that.
Yeah, I mean, I couldn't agree more in that, we have to use the in vivo models that are out there to answer a very specific question. So whether that be toxicity issues, but when it comes to really disease-specific mechanisms, which quite often for a lot of these neurodegenerative diseases are human, they're very specific to human molecular machinery. So there we essentially have to take a step back from the in vivo models, go back to in my opinion, go back to the human models, and use those to understand both on-target, off-target effects, and then work our way back up into the human. And again, I do think in vivo models do have a purpose, but it's going to be more for the safety perspective, and potentially some disease mechanisms may be translatable. But as long as we're acknowledging that it's not the human system and we understand the caveats associated with that assumption, then I think we would be in a better position.
Astrid, I know gene therapy has never had an issue translating from animal models to humans, so I don't think you'd have anything to say on this topic, right?
Thank you for the challenging question. No, I think I'm in between of what Ross and Shu are saying. I do see the value of all the models actually, because only then, when you have the right in vitro models, when you have the right in vivo models, you can have a guess of whether indeed it has a chance to work in the clinic, especially in terms of CNS diseases. Because one of the challenges that we have that I know there are a lot of models, but still crossing the blood-brain barrier is an issue. Distributing to the right brain areas is an issue. Targeting the right cell types is an issue. Interaction between cell types, you can model it in vitro, but part of it, I think, not in the totality of it. And you would agree with me that some cells behave the same, similarly in vitro than in vivo, but not always. It's a hard time to model right situations, and I know, there are lots of advances in this space. So to us and to me, it's the totality of all of these models. It's more work, but I think we owe this to the patients. We want to bring to the patients something safe, for sure, but something that has a chance to be efficacious. So how we work it out at uniQure in this case is in vitro models, rodent models, and a few large animal models, not only for tox, but also for biodistribution, and show that your therapy is going to reach the brain in the right amounts, and it's not going to reach other organs that you don't want to reach. We're not there yet to avoid these studies, although we need to be very critical when we do them, from the ethical point of view, of course, as well. But I think it's key for the CNS. And I think that some of the therapies that have failed in the clinic is because most likely they have not reached the brain in the right amounts and have not reached the brain cells that they would need to reach. So learning more about that, and going back to what Ross was mentioning about having these preclinical animals models being predictive of what we will see in the patients is key. Also, when in terms of biomarkers, we measure biomarkers in the biofluids, which is great. We can measure brain molecules in the biofluid, but what does this mean? Very often we don't know, the source of these molecules, because they're very often also expressed in the periphery, and it's difficult to interpret these changes in humans. So we do need in vitro and in vivo models to try to figure this out and interpret much better what we see in patients. So in my view on this.
Thank you. Rebecca, I know you spend more time probably with patients than with gloves on, or I guess probably have gloves on with patients as well, but less maybe time with in a research setting and more time in front of the people actually suffering with the diseases that we're talking about today. I mean, from your perspective, and we'll come back to this translation gap question because I think it's a really interesting one, and we've had a question kind of in that vein as well. But I wanted to kind of ask you, because if do you feel like these clinical trials are asking the right questions?
Yeah, so I think that that's a great question. I mean, there's been so many failures, and that of course is very dejecting to people with Parkinson's. So a couple things that I often hear from people is, because we talk about how much variety there is, we love to say as movement disorder physicians, if you've seen one person with Parkinson's, you've seen one person with Parkinson's, very similar to what Dr. Ross said earlier. And so do we really expect one drug to be effective for everyone? That is contradictory. So perhaps our clinical trials need to be we need to be more splitters than lumpers, and find the people who the drug does work for and not just declare it a failure. So I hear that a lot, and I do think that is something very valuable. Should we be going back to failed clinical trials and finding, is there a signal somewhere that can be pursued? So that that's something to consider. And the other thing to consider is people understand that we're not necessarily going to get a cure, but perhaps we can get an improvement, and that could be huge too. Now, of course, we don't want to be introducing a product into people that's going to cause brain inflammation or something very dangerous, and that that of course is something that we're all very aware of. However, if our goal isn't necessarily we have proven that progression has stopped by 20%. What if it's just I can spend more time with my grandkids, or I can take care of my grandkids on my own, or some readout like that would be huge for some people. So I think we have to, again, Dr. Ross was saying readouts and how do we really quantify that, but thinking about the patient perspective, finding readouts that are meaningful and our quality of life encompassing, I think could be, a way to think about this as well.
Yeah, I think it's one of the things that I'd be quite interested to hear y'all's thoughts on of are we dealing with good therapies in bad trials? Bad therapies in bad trials, or bad therapies in good trials? And then, are there lessons learned from kind of the oncology space where, there was a ton of confusion, right? When the first sort of surface markers on solid proteins and the first real inhibitor classes of drugs where you started really doing heavy typing of the, well, a friend of mine with a diagnosis was relieved when it was PD-1 positive, right? I can't imagine, do you see that getting to a similar state of 101 flavors to maybe be a little bit, well, a little bit non-scientific here? Do you think, Shui, do you think we've had good therapies kind of slip through our fingers because the trials didn't catch the right patients, or maybe we're not even measuring the right things, as Rebecca was alluding to, in terms of behavioral and quality of life? Like, where do you fall on that sort of, have we seen a good therapy already and just not known it, or are we still hunting?
Yeah, I think it's going to the answer's probably going to be both. There's going to be therapies which may have worked, and that could have been for a number of reasons. It was brought up earlier that we quite often these clinical trials, these patients have pronounced neurodegenerative neurodegeneration, and we're just going in too late. So they could have been effective if we indeed were able to diagnose them earlier and intervene earlier. And then indeed there'll be therapeutics which are going to be more efficacious in specific individuals, whether that be because they have a specific genetic makeup or been exposed to certain environmental factors. So there we do. I'm not a statistician or a data scientist in any way, but I think it would be very, very useful, an important step to go back through old clinical data to see if there is a hint that we could repurpose an already failed drug.
Astrid, maybe I'd love to get your thoughts on that, given sometimes
Yes.
clinical endpoints in gene therapy can be oversimplified, shall we say. Do
Well,
you see
they're
the
not
same
always.
chance that there's been a winning therapy that's passed through, because of the wrong data interpretation, or do you think it's a little bit maybe more straightforward?
Depends very much on the indication, I think. For some gene therapies, it's a little bit easier. If you take hemophilia and you have factor 9 as a readout, and the patients don't need to go to the hospital when so often as before. It sounds a little bit more simple. It's not, but it's a little bit more straightforward that for Huntington's, for instance, which is like all neurodegenerative diseases, a slowly progressive disease. It's heterogeneous. And of course, we have the same challenges with the clinical readouts, which is variation. So I would agree very much with what Rebecca was mentioning about looking back at the data, and this brought to me the idea of the concept of data sharing. So there are, very valuable consortia in which data sharing from different stakeholders is stimulated, not only to know whether trials have failed, but also to build a very solid natural history, which we need for many of these diseases. I think that this is the basis, right? And in many of the trials, less in gene therapy than in other modalities, but we have a placebo control arm, which we can, leverage and share with others. And if there would be more agreement about which readouts within these patients, we will build as a community a very solid natural history, which can be a very good comparator maybe in the future, right? So it would help us find out these patients with populations a little bit better. It would help us know, which clinical readouts, which biomarkers might be helpful to pick up signals. So I think this is something that in this space, there are more and more of these initiatives, which I think is key. We need to build therapies that are going to be transformational. At the same time, the patient needs to feel the added value. Again, alluding to what Rebecca was saying, they're the ones that are able to say, if their quality of life has improved or remained stable. And it would be great to have also patient-centric readouts because, it's not numbers, it's about they feeling better. So I think that's also where the space needs to move forward.
Rebecca, I'm interested, I heard a couple of things in what Astrid was saying that that I'm familiar with as being led by patient advocacy in some ways, right? Whether it's consolidation around an endpoint, consolidation around things like natural history studies, and certainly for trying to frankly break down the walls of pretending like some things are proprietary when they probably shouldn't be, right? To help us all move forward. Do you, from your perspective, do you see those efforts as successful, or is it kind of always trying to break down some walls as we're working towards a common goal?
So I do think that the Parkinson's world is working together a lot more than in the past. And I think there has, been a push, in our move to include the patient voice more and more. I think that is something that comes across loud and clear from the patient perspective. Don't tell me that the reason that this isn't moving forward is because you're not talking to each other. That's unacceptable. And I think people understand that. So I do think there's been a lot of value on that particular point with including the patient voice. And we should also say that in these diseases where genes certainly play a role, they're not the whole story. Clearly in Parkinson's, they're not the whole story, and Alzheimer's are not the whole story, but there certainly are genetic elements to some of these diseases. You have the families, who are very motivated. And sometimes you have the families who carry the gene. So for example, in Parkinson's, there's one of the genes is LRRK2, which you can inherit, but you're not necessarily going to get, you only have a 30% chance of getting Parkinson's with the inherited gene. So you have this population where they're motivated. They may get it, they may not. They're sort of in limbo. And this is a very motivated population. They're seeing the effects of the disease. They carry the gene. And so these are people that, have become involved, have been leveraged. And as I was saying, earlier, in terms of moving clinical trials to a more early state in the disease in order to potentially intervene, that population is going to become important to actually potentially even join clinical trials at some point. We're not up to that yet, but that's sort of could be the goal. So I do think that there's been a lot of movement and change over the last few years in these issues. So I think that's really a good thing.
Ross, I'd be curious to see, and I'm leading to a question here that's been posed by one of our attendees. I think you sit astride this sort of connection between the patient and some of the science directly. I'm really curious how you think about, as an example, connecting research and development efforts to hanging out with my grandkids more. Like that's what the medicine's for, right? Ultimately. But I'm trying to think about sort of similar to this translation gap issue that we talked about before, and how do you have an in vitro development group that understands that's the objective, and how do you connect the physician and the patient to defining the problem statement better for that researcher, whether they're in academia or whether there's somebody in your group at Immuneering, right? Like how do you think about that, spending time on both sides of the fence, so to speak, and I guess how high is the fence is a good question in of itself.
Yeah, that's a really good question. When I was training as a neuroscientist, and maybe this is was a sort of a fad. We placed a lot of emphasis on bench to bedside, sort of hearing stories from the clinicians, from the patients, and sort of getting their perspective of what they're going through. And one of the things I've sort of learned through my experience with working with patients, including my father also passed away with Alzheimer's, is that the sort of the symptomology of it is very complex. Like you have folks who have insomnia, you have folks who have a little psychosis. And so the question becomes, how do you sort of tease out those patients within the data that you're getting from the patients? And so for my researchers, I always place an emphasis on, how does this connect back to the disease signal that we've isolated from patients that have these sort of symptoms, and whether or not that would translate into a particular target. That's another thing we really haven't talked about here on the call is what are we doing to sort of identify targets that are specific to these subgroups, and whether or not that those targets are related to some of the disease progression we see in those patient populations. So, for example, we know that with Alzheimer's, there are just sort of different flavors of atrophy, where you have sporadic brain loss throughout different brain regions. You sometimes have hippocampal sparing. How does that sort of translate back to sort of the transcriptomic data that we have? For example, is there something that's unique to those patients? And ultimately, that's going to translate to things like working memory, time perception, et cetera, et cetera. So I think these are the kind of deep questions that we need to sort of take a step back and reassess as a community, a scientific community, because this whole idea that there's just one disease and we're just recruiting people with this one disease to the trial, it's definitely not the right way to go, the most efficient way to go.
Thank you. Shui, I'd get your perspective on kind of two different questions the audience has posed here at once that I think we're moving towards in an interesting way in the conversation here. Ross and Rebecca both said, Astrid's mentioned as well, that there's even in very clear mechanisms of action that may be, genetic cause, have a genetic cause at the root, there's still multiple subtypes. I know you spend a lot of your time creating disease models that are from patients. How many of those do you think we need before, before we capture a meaningful percentage of the population? And then more on the infrastructure side, how many of those do we create before it's in effect a bespoke therapy? Do we, if we move towards this sort of, in your case, Shui, I know it's really iPSC focused, but if we have 17 different iPSC models of Alzheimer's, do we have 17 patients worth of therapies at that point? Do we have the infrastructure to view those diseases that way yet, or are we still kind of behind on technology to answer those questions effectively?
I think, from an ambition perspective, we certainly want to put them into bigger buckets that would just expedite the drug discovery process and also getting the therapy to the patient. I think we're beginning to have the technology to finally discriminate between AD populations. Oh, sorry, not AD populations, but individuals with Alzheimer's disease and potentially subclasses of Alzheimer's disease. But here it's very important that we begin to link genetic data and phenotypic data, so at the in vitro level, with what the clinician is seeing. So just as and we're not able to, for certain memory-related phenotypes, we're not able to, effectively model that in a dish, but there could be signatures that we can pick out and link them to a clinical event. So there, I think there needs to be more information sharing between physicians or healthcare systems to scientists or biotechs so that we can actually begin to make this link for the entirety of the day. If we have to head down the route of a personalized medicine, for these diseases, I mean, I'll be honest, I think it is going to be a challenge and there will be hurdles along the way to be able to do that. But I think for the, in the, rare and orphan disease space, it's been clearly shown that this is possible. These are still small scale, and certainly with antisense oligos, you can really go from a single mutation to a patient with a tailored medication in a very short amount of time. Now, it's the beginning, and we have a long way to be able to translate that to diseases or neurodegenerative diseases with such prevalence, but we are beginning that journey.
Well, Astrid, I'd love to get your kind of perspective, because you've been working, the uniQure Huntington's program is borderline, considered rare by most definitions, even though it may not be the rarest rare disease, right? But when you're looking at development, how do you see the experience in gene therapy translating to these larger modalities, both in terms of the regulatory pathway, where I think there's been a lot of acceleration? I think, for just on a personal level, it's been interesting to see and be a part of a couple of gene therapy registrations that seem to happen in 5 or 6 years. I mean, literally, they did happen in 5 or 6 years, while, non-gene therapy programs with maybe a better understood modality are moving much more slowly. When you and your team there are thinking about translating the learnings, where do you shake out on that? If everything needed a pretty bespoke genetic medicine, are you looking forward to 17 parallel clinical submissions, or do you have to go higher level than that for this to be feasible?
Well, at this moment in time, what I was trying to mention before, even in the space of Huntington's disease field, it's a rare disease, but it's also underdiagnosed for sure. And we are not talking about an ultra-rare disease. And being monogenic, still complex, and there's still subpopulations. What have we learned in this space, and what are the challenges, and how can this help in Alzheimer's or Parkinson's? I think what we are starting to learn is that maybe for a single target, different modalities can have different challenges. Huntington's disease field has gone, very much focused lowering the levels of huntingtin and mutant huntingtin. But there are different ways you can do that with the pros and the cons, and we are learning from each other. And actually, it's a field that has had quite a lot of drawbacks, unfortunately, during the last year, with many trials being failed for different reasons. And I think, you know, something that is excellent in this community, Huntington's disease community, is that as much as possible within what we can do is the data are shared on time, so that we can learn from what didn't go fine, right? And try to understand that. And this is key. And I think that this could be valuable to many other indications in the neurodegenerative space. So what we learned from the trials that failed, and it's not that we stop and we don't anything with the data. We analyze the data as good as we can, and we share it with the community, we discuss it, we put it out there to make better therapies. Because sometimes it's not that the target is not valuable. You know, Huntington, this is the target for Huntington's disease, many others as well, of course, because it has many interactors. But you know, how much do you need to lower it? Where do you need to lower it? When do you need to lower it? Which are the readouts that you need to have in the patients, etc., etc. These are the kind of things that we are trying to learn, and I think it's going to be informative for the broader community. So yeah, that's a little bit what we are talking with right now, and why we as a company and other companies are also moving forward in the more broader indications in the neurodegenerative space as we move forward.
I second that point on data sharing, and that's why I want to give a shout out to organizations like AMP-AD, ADNI, where we sort of build a sort of collective of being able to feel comfortable sharing data, because ultimately, sort of like what Shu is saying, this ability sort of to integrate data, and to be able to present it to the community, make it more accessible, get out of our silos, is the only way that we're going to be able to move forward with and make progress on these very complex diseases. But I have a question for you, Shui, to sort of go back to Andy's point. How many stem cells sort of do we need? You and I know, sort of like from personal experience, these are very costly experiments to do. And it feels, in some sense, that every time we do these sort of exercises, it's like building a car, like one car. How do we get to the point where we're like, have the Henry Ford model, where we have sort of a more scale up infrastructure to really interrogate a number of patients instead of just a selected few? Where do you see that going, Shui?
I think this is, some, just to delve into the science and the technical a bit here. There's some great work from a group in the UK, which is beginning to understand what drives, the challenges in getting in vitro models produced robustly, but across a broad range of patients. And there, I think it's important that we, control the process and the material that is entering into producing these complex cell models. And there, I think we can indeed, switch over from a really bespoke per patient approach to using panels, of different iPSC lines that are well characterized, both from a genetic perspective, but also from a phenotypic perspective, and then get them to the correct cell type. So again, I don't think we're quite there yet, but indeed both the understanding at the iPSC stage, but also the processes to differentiate them, those are all getting more and more robust. And it's a collaborative approach between, companies like ourselves, but also academia, who are doing a great job in teasing out the really key details.
Rebecca, I'm kind of interested in hearing, when you hear kind of the development and streamlining of those sort of toolkits that are disease specific. Where does patient advocacy sit in owning and giving access to those sort of tools, as opposed to them being I'm just trying to reconcile the Ross's comment about siloing your efforts, which I think are very successful in helping connect different what would be competitors in the right light, versus the expense and the complexity of creating those models. Warehousing those things so we're all speaking the same language, so to speak, would be incredibly useful, but it's also, time and money are finite. Where do you see the utility of those tools as potentially providing them to industry as patient advocates?
Yeah, so that's a great question. I mean, we all understand that the profit motive is huge, and meaning it's important. It's an important part of this. We need there to be a profit motive so that people want to work on it. That's just how capitalism works, and that's understandable, and everyone acknowledges that. So in order to de-risk that and to encourage things that aren't necessarily obvious benefactors in capitalism, that's where the nonprofits can really play a huge role, because the money that we have is able to be funneled to different organizations, not based on who's doing the best in the market, but who we think can really move the needle forward for the patient. So I think a combination of industry sort of being guided by the patient voice through nonprofits is really a successful way to deal with the issues that you're bringing up. So I do think there's a really vital role for the nonprofit world, more today than ever, I think. Thank
you. I know we're 4 minutes left, and our 4 awesome panelists are here. So I thought I would end by kind of giving you each 60 seconds to answer the question I forgot to ask, right, that you really wanted to talk about today. I've taken a lot away from this in terms of the biomarkers that are needed, kind of the tools around translation, all the way from the academic benchtop to the clinic, which is a long way to translate. And then a lot of takeaways around communication and connection between the stakeholders as the key pieces here, because I don't think anybody here seems convinced we're asking the right questions yet at the right times to have meaningful answers for large segments of these populations, given the complexities of the diseases. So those are certainly my takeaways. I hope you guys enjoyed it. Astrid, go first, and what should I have asked that I didn't, and what do you think about that topic?
You didn't ask me that you would ask me about that. You didn't tell me that you would ask me about that.
But, that's the nice part about having smart people on a panel, is if you put them on the spot, you're going to hear something interesting.
Yeah, sure. Yeah, it's a difficult one, but yeah, maybe we talked about a lot about what we need to change, right, to make something that really works for the patients. And I think I think that the answer is what we all point to, which is, put all the stakeholders together and have an holistic view to be able to create therapies that are going to change the life of these patients. So I think this is a mindset change that is taking place already in the disease space. But being aware of it and being very active in making it possible is the only way of changing the situation that we are in now. So holistic view, working together, and take all the points of view into account. But yeah, long answer.
That's great. Ross, any closing thoughts?
No, outside of full disclosure, I'm a board member for the Alzheimer's Association, so I just want to give a shout out to any patients on this call or watching this recording. Please reach out to Alzheimer's Association. They have a lot of great resources there. But I think one of the things too that's sort of a deficit in our field is just patient samples. We need more patient samples. The more patient samples that we have, the better we can sort of infer treatments, targets, biomarkers. So real proponent of that.
Thank you. Shui?
Yep, thanks. I was hoping to be left last. I was still thinking. I think really the question not only for the panel but also for the people listening now, but also in the recording, is what can we do to facilitate and move things forward? What is the biggest hurdle that we're facing? You know, we've gone from in vitro and vivo to clinical trial setups and readouts. What can we do in the short term to begin to tackle some of these problems? And maybe we have some of the answers, or in terms of just enhancing the collaboration, data sharing, but I'm sure that's not going to be the be all of it. So yeah, that would be my next question if we could go on longer.
Thank you. And Rebecca, I'll give you the last word as a panelist here, if you don't mind.
Thanks. Yeah, so for me, the thing that wasn't brought up, which I think we should mention at the last moment, is testing lifestyle changes. So that's something that, you know, industry isn't really focused on, but diet, exercise, you know, we have data that those things are super important, and we probably get a lot out of that. And pushing that agenda forward, I think, will be hugely beneficial for everyone. So want to put a plug in for those type of thoughts as well.
Thank you. I was tempted to bring up epigenetic changes caused by diet during as a result of one of the questions, but resisted because I thought everybody was already sweating enough when I put you on the spot here at the end. Thank you again. Thank you to everybody who attended. For anybody who submitted questions, I'm going to actually pose those to the panel just in case there were any written responses that they didn't feel like I did a good enough job to give you guys a chance to answer. There's also one final question right at the end that I'll just throw to the panel as well that we didn't get quite enough time to address. But thank you again to our panelists. This is a lot of time that they have spent to be here today. I certainly found it educating. I've got 2.5 pages of notes of smart things y'all said that I'm going to try to remember, and I hope everybody who came felt the same. Any feedback from the panelists or for any attendees about how we can do these better, I'd love to hear, because these conversations are a lot more interesting to me than you know, other formats that may be less interactive or more shall we say product focused. So I really appreciate everybody's time and attention. Thanks again, and I hope you have a great rest of the day. Thanks.
Thank you.
Thanks, everyone.
Thank you very much. Goodbye.
Bye-bye.
Bye.